and welcome to this Waterloo Smarter Health Seminar. I'm Shirley Fenton, Managing Director of the Waterloo Institute for Health Informatics Research. Thank you for joining us today for our second seminar in this series entitled The Many Facets and Faces in Health Informatics. I'd like to welcome our web viewers and the Ontario Telehealth Network sites. This seminar is being recorded and is being broadcast over the web. We ask that you hold your questions to the end of the seminar presentation so that we can get a real or virtual mic to you. A very warm welcome to our guest speaker, Dr. Richard Kim. Richard is a professor at the University of Western Ontario, a scientist at the Lawson Health Research Institute. He's also the chair of the Division of Clinical Pharmacology and director of the Center of Clinical Investigation and Therapeutics at the University of Western Ontario. Many, many titles. Dominic will introduce Richard more fully shortly. I'd like to acknowledge our seminar series sponsors, Borden Landner Gervais, McKesson, and Healthcare Information Management and Communications Canada. Without these sponsors, we could not host these seminars, so we're very grateful for their support. Before we start, I'd like to announce our next seminar in this series. It will be held on Wednesday, March 26th, and our guest speaker will be Mr. Bill Albino, who is the CEO of Smart Systems for Health Agency. The title of his talk is Smart Systems for Health Agency, Helping Improve Ontario's e-health care through e-health innovation. This new seminar series will look at the many facets and different facets of health informatics, as I said, and some of the key people involved in these areas. Over the next several months, our guest speakers will talk about personalized medicine, medical imaging, information and communications technology to support our healthcare, and the role of geographic information systems in healthcare. So it's quite a broad range of topics that we're looking at, and I invite you to come to each and every seminar. So our agenda today is as follows. Dominic Covey, Founding Director of the Waterloo Institute for Health Informatics Research, will introduce our speaker. Dr. Kim will speak for about 45 or 50 minutes. We'll then have a facilitated discussion. We ask that you hold your questions to after his presentation, and we'll get a mic to you if you have a question to ask. Just a reminder for those of you that do get a mic, please position it as close as you can so that we can record your question properly. So may I now introduce to you Dominic Covey, and he will introduce our speaker.
Which end of the mic, Shirley? Is it? She always talks about the mic because she has such trouble with it herself. So first of all, welcome to you, and welcome, Richard. We're really glad to have you here. Shirley's made the major points about Dr. Kim's background and so on. Right now, Dr. Kim is based at the University of Western Ontario, and I only really learned about you about a year ago through Jake Teason and others that we were speaking with. He came and was brought here along with members of his group from Vanderbilt University, and I cannot tell you what a coup this is for Canada and particularly this area. It gives us a potential that did not exist. It was literally imported. I presume it was under the NAFTA agreement, that very little in the way of taxes and import duties, I hope. And he's established a significant group there. We're talking about his new facilities, which are going to be completed relatively soon, about 7,000 square feet. I think he was the biggest purchase made by the University of Western Ontario in its history. I think what really excites me about this is an absolute commitment. What do you say? They bet the farm, I think is the term used. The absolute commitment of a university to an area and a group of people with really an incredible track record. It's really great to see that, the investment in people. I know we buy lots and lots of equipment and facilities, and millions and tens or even hundreds of millions go into CFIs. But the idea of bringing people here, which is around, which need to be there around and build research around them, it isn't the equipment, it isn't the facilities, it's the people. And I think we're incredibly fortunate to be able to get this group of people, particularly you. I also point out that the Institute is planning to work very closely with Richard and his work. bringing aspects of health informatics and other capabilities at the University of Waterloo, including bioinformatics and engineering. So we have been invited into, and we're very honored to be invited into, a very significant collaboration. And we look forward to really building that out over the next several months. And when David Cobb speaks, we'll talk about yet another collaboration that's finally come to fruition, which is a very, very large undertaking as well. So, Richard, welcome. We're glad they got you here, and we're glad to have you here. Thank you.
So first of all, I'd like to thank Dominic and Shirley. I've had a great time visiting Waterloo, and drive here is actually quite pleasant, and it certainly beats going to the airport. So delighted to be here, and also delighted to have a chance to go over with you some of our research and some of our focus in terms of personalized medicine, and hopefully to spark some interest in many of you regarding what could be done, and also to kind of impart with you this notion that to make this area of research really meaningful to the delivery of care to our patients, that we do need to really think about the effective collaborations and synergies in terms of skills that we have, because I don't think any one group has a monopoly on this field. So I do have a lot of titles, and I showed them again, but we'll move forward. So I do want to kind of let you know that actually I didn't just come from Vanderbilt, but in fact I did spend a lot of time in Saskatoon. I grew up there. And I always show this slide. People in Ontario sometimes forget where Saskatoon is. So it's near Winnipeg and Calgary. And I think all of us from Saskatchewan are hardworking people. Fortunately, I only had to walk 10 miles a day to school, unlike my classmates who did 50. And this is a good day. So, of course, we went to a very fancy school, and I don't have a problem sharing that with you. But, of course, I'm exaggerating. This is Saskatoon as is today, and it's a vibrant city, and not unlike Waterloo. It's really a university town, and as of late has done very well given the resources and all the activities going on in Western Canada. So one of the things I've found that in the past, certainly a year or two that I've been here, is to really kind of let people know not only that we do personalized medicine, but to maybe convey to you we are relatively serious in terms of our abilities, and that we're not just sort of jumping on this bandwagon of personalized medicine. So I'll just give you a little bit of a sense as to who I am, who our group members are, and what it is that we do. So I do have a medical degree. I did internship and then a residency in internal medicine in Saskatoon. And then summer of 1991, I went to Vanderbilt University to do postdoctoral fellowship training in clinical pharmacology. This is a picture actually from 91. My daughter, who's currently going to university in Toronto, that's her 2 years old. We packed everything into our station wagon, sold our house, and left. So we actually drove all the way from Saskatoon to Nashville, Tennessee, where Vanderbilt University is located. And at Vanderbilt, I know some of you have been there or know of it. It's really a vibrant and excellent medical center. If you have a chance, you certainly should visit the place, and really a leader in many aspects of medical research as well as patient care. So this is actually a picture from my group from 2005. So we actually did grow into a fairly large group, and we did actually move to the University of Western Ontario in 2006. And I was fortunate enough to be able to take a couple of faculty members as well as postdoctoral fellows. So here you see two of my faculty members as well as 3 postdocs moved with me. So we packed everybody up, and the car floats over the lake, and we've been able to get to London. So I don't often show these slides, but I'm trying to get as many brownie points as I can. So it turns out after I moved to London, we must have done okay. So this is a London Free Press. This is probably not the circulation that Globe and Mail has, but I made the city section, and it turns out I'm a medical star. Shortly after that, I made the top 10 sexiest men of London list. But if you look at the fine print, actually, it says 10 people to watch. So I was kind of darn wrong list. So let's start. So our group has been interested in this notion of intersubject variability and drug responsiveness. And I showed this picture primarily because, as a physician, and when you see physicians, you probably do wonder how do physicians come up with the drug and the dose. And it's actually not because of how you look, how old you are. We actually have no a priori way of knowing what dose of a drug or what drug would be best for you. So we typically look at your symptoms and come up with a class of drugs that may work for you, and typically prescribe an average dose of a drug. So this is a slide that illustrates some of the current thinking that may be different than what you're thinking. So this is an example where it shows if you're able to measure blood levels of drugs, this actually shows that this Neanderthal man actually has the highest blood level versus the little kid who has the lowest blood level of the drug. So this says to us that, in fact, if the blood levels predict drug toxicity, in fact, the Neanderthal-looking man may be the most susceptible to drug toxicity. And it really goes against our notion that just because you're a big and hulky, strong-looking person, that you should be really strong against medicines. It may not be the case. So our group has been focused on drug levels and efficacy and really considering all of the facets of environment, disease, and genetics. And of course, underlying these are really what makes a drug disposition different. So these are processes that govern how drugs are broken down or metabolism, as well as how drugs are moving in and out of the body, known as drug transporters. And of course, these transporters and metabolizing enzymes are regulated by the body so that some people have lots of metabolism and lots of transport. Some people have very little of transport or metabolism. So I just want to give you a little bit of sense as to that. In fact, we do have some credentials. I'm going to show you some of our classic hits, I suppose, or papers to show you that we have fairly diverse interests, and we've done reasonably okay in terms of the type of studies we've conducted. So this was a paper from our group from about 10 years ago, published in JCI, that actually said there's this transporter protein called P-glycoprotein that, in fact, was a major limiting factor to the absorption as well as the CNS penetration of an important class of drugs known as HIV protease inhibitors. So around 10 years ago, protease inhibitors became really the profound drug that changed a course of HIV therapy. In the 90s, there was a lot of desperation and I guess distraught over people who were dying from AIDS. And when these class of HIV protease inhibitors were released, HIV became a manageable chronic illness. But we knew even then that the drugs that were efficacious still was not fully efficacious because they were unable to get to all sites of action, including, for example, the brain. So I'll show you a little bit why this was an important publication. So what we were able to do is we knew blood-brain barrier is importantly regulated by this transporter called P-glycoprotein. So we took a mice model. So this is a mouse lacking this gene called MDR1A. It's a P-glycoprotein deficient mice versus the wild-type mice. And this figure shows you sort of the ratio of the knockout mice versus the wild-type mice. So for the three drugs that we studied, indinavir, nelfinavir, and saquinavir, the knockout mouse to wild-type mice ratio of drug levels were quite similar in most of the organs, except in the brain. In the brain, the levels of these HIV protease inhibitors were 7 to 40 times higher in the P-glycoprotein deficient mice, suggesting that, in fact, this was an important barrier to the CNS entry of drugs. We also have a significant interest in understanding drug disposition gene regulation. Why do we have this interest? We know our genetics have important impact on how we handle drugs, but we know at any given time, depending on what you're exposed to in the environment, what kind of other medicines you take, as well as dietary constituents, you can actually change the express levels of the proteins that break down or transport drugs. So this is what is typically referred to, at least for our purposes, transcriptional activation of genes by nuclear receptors. So I'm just going to do a little bit of teaching for you. It may or may not be quite understandable, but one way of increasing gene expression in the body, especially for drug metabolism, is for drugs, and it could be called a ligand, would interact with a nuclear receptor, and then nuclear receptor would get into the nucleus and actually transactivate or increase mRNA synthesis, which then becomes lots of protein synthesis. So this is a way for your body to rev up expression of target proteins, and if you express lots of drug metabolizing enzymes, you can imagine it would break down drugs more effectively. So this is our publication that we had a couple of years back now that showed that, in fact, this orphan nuclear receptor called HNF4 alpha was a major determinant of other nuclear receptors called PXR and CAR in the induction of a drug metabolizing enzyme called CYP3A4. So I just give you a sense as to the kind of things we do, but not necessarily really fully go into this topic. We have also studied drug targets. So we are interested in fundamental biology, but we're also very much interested in clinical relevance. So this is a publication in which our group was one of the members that was published in New England about 7 years back now that stated that polymorphisms in the beta-2 adrenergic receptor was very important for vascular sensitivity. So at that time, there was a lot of activity going on about a couple of these genetic variations. But what we were able to do was actually show that some of these variations occurred together. So this is this notion of haplotypes, and that certain haplotypes were actually more predictive or important to predicting vascular response mediated by beta-2 receptor activation. So, having confused you a little bit about the kind of things we do, I hope I've convinced you that we don't just look pretty, but we actually can do some science. So now I'm going to talk about more of the warm and fuzzy side of personalized medicine to give you more of an overview of what the field is doing, and then also end with where we need to go, both within our group as well as within all of us in terms of collaborations that would foster movement in this area in terms of something that actually impacts patient care. So I need to reiterate that variation in drug response is this interplay between environmental factors as well as disease states and genetic variation. So these are all the factors that we need to consider as we want to individualize or personalize therapy, because you can imagine we're all born with the same genetic makeup, but from birth to our old age, you know we're going to vary in our ability to handle or respond to drugs. So it can't be just our genetics that control all of our variation. So with personalized medicine, the hope or the goal is to really avoid toxicity, drug interactions, predict responders, and then, if possible, targeted drug delivery and certain kind of areas such as cancer to really individualize therapies. So just a couple of examples. I think a lot of people cite this, that adverse drug reaction is the 4th leading cause of death among hospitalized patients. We always think we go to the hospital to get medicines, but we sometimes forget that, in fact, medicines themselves may be a major cause of mortality in our hospitals. So that is suggesting that, in fact, either we're using the wrong medicines or medication errors, allergic reactions, or wrong doses of medicine. So, in fact, this is a huge problem that affects us. And there are simple things that many people talk about, that, in fact, 1 in 11 patients will not benefit from drugs like codeine, no matter what the dose, due to genetic defect in an enzyme called CYP2D6. So a number of you in the audience probably have this defect. So what people don't typically understand is that drugs like codeine, which is widely used as a pain reliever or analgesic agent, itself has no pain-relieving properties. It is actually bioactivated or converted to morphine by this enzyme called CYP2D6. So if you happen to be genetically deficient in 2D6, you would make no morphine, so no amount of codeine will help you. Lately, there's been additional interest in this medication that's widely used to treat breast cancer called tamoxifen. It turns out CYP2D6 actually can generate active metabolites of tamoxifen that is actually far more potent than tamoxifen itself. So there is some concern that, in fact, people with the 2D6 deficiency may not benefit as much from tamoxifen for breast cancer therapy compared to those with regular functioning 2D6. And of course, we're always hoping that new pharmaceuticals will save us, because I think we believe high throughput screening and target selectivity and potency will save us from having to deal with old school drugs that are given in large doses that are not as selective. But I think by and large, we have been disappointed by the newer drugs, because you hear about things like this. So one drug that was recently pulled off the market for irritable bowel syndrome called Zelnorm, this was only about a year ago that the drug was pulled off the market, because it did more harm than good. There's been a lot of publicity and discussion about this important class of drug to treat diabetes. This is a drug called Avandia, or rosiglitazone. As you know, there's a lot of concern that this class of drugs may actually cause more cardiac problems than necessary, and whether it's really worth the risk. Another drug only in November of last year pulled off the market that also again suggests problems with some of the newer drugs that are being marketed. So intersubject variability in drug response could be reiterated again in this format. You take two people that look very identical. So I always tell the joke, two transparent looking people, as you can see right through them, like some of your colleagues. But if you could actually measure blood levels, you might actually see that the first person had a very high drug level. And we don't typically measure drug levels, because often it's viewed as difficult or onerous, but we do measure things like blood pressure. So if this were a blood pressure lowering or antihypertensive medication, you might see that, in fact, the first person might have a profound drop in his systolic blood pressure, perhaps sufficient to end up in the emergency room, whereas the second person that had a normal drug level, in fact, had an okay response and did not require any intervention. So the current practice of treating our patients is really this iterative process. So I think all of you in the audience and those on the web could recognize this practice. So let's say you have a cold or you're coughing up stuff. What do? You go to your family doc. And I think most of us would agree it's almost a cultural custom. We will not leave the doctor's office without a prescription. So when you get the prescription, it's almost a cue for you to leave. So when you get the prescription, you feel it, you take it, three things can happen. It can be effective, which is great. It can be not effective, or it can cause some side effects or toxicity. So when you have lack of efficacy or toxicity, what do? We go back to the physician, and then he or she may prescribe a slightly different medication for you or adjust the dose. And this is an iterative process, so you have to do this over and over until you obtain benefit from that class of medication. So personalized medicine is really then trying to go over this notion of the interplay between our genetics, the environment, and our diet, as well as our clinical condition. So preemptively then come up with a drug that may work better for you, both the right drug as well as the right dose. So in this case, you can see that maybe drug A, it could be drug B, or it could be half the dose of drug A. We have the human genome fully sequenced for many years now. And of course, there's a huge race on to try to sequence the human genome again for $1,000 or less. So the technology is moving at a tremendous pace, because the first genome costed billions of dollars to sequence. And there's a lot of perhaps hype as well as hope that the genetics may solve a lot of our problems in terms of health disease. So a little bit about background information about pharmacogenomics for the students in the audience. The human genome diversity, we think there are billions of nucleotides or base pairs. The human body probably has in the order of maybe 24,000 genes. All of us are 99.9% identical at the genetic level, but it's that 0.1% variation. that defines our individualness, I suppose. And that variation also impacts our ability to break down or handle drugs. So these variations are often referred to as SNPs, or Single Nucleotide Polymorphisms. So let's talk a little bit about drug metabolizing enzymes and adverse reactions. So again, for some of you who don't really think about how drugs are broken down by the body, a little bit of, chemistry or biochemistry 101 for you. The human body, certainly the human liver, does not have thousands of enzymes that break down thousands of chemicals. It actually uses only a handful of enzymes to break down all of the chemicals that we ingest, including drugs. So if you if you took all of the drugs that we prescribe today and asked the question, which enzyme breaks down that drug, it turns out, in fact, a handful of these enzymes do bulk of the work. So CYP3A4 alone, this is one, drug metabolizing enzyme alone is thought to be involved in the breakdown of nearly 50% of all the drugs that we prescribe. And other CYP enzymes, including CYP2D6, 2C9, also are very important. And some of these CYP enzymes have major genetic variations, and we think contributes. And I'll show you some data that are important for a lack of drug efficacy or toxicity. So this is a slide that illustrates our notion of understanding genetic variation even before these genes were cloned or identified at the molecular level. So this is a drug called debrizoquin, which was developed as an antihypertensive agent in the early 70s. And during the early clinical studies, it was noted that approximately 1 in 10 or so subjects appeared to have a profound reduction in blood pressure. And the other people seem to be quite normal. And when they actually systematically studied those subjects, they noticed that, in fact, there were two populations of people. The ones that are referred to in this slide as poor metabolizers, or PM, or the extensive metabolizers, or EM, which would be considered kind of a normal phenotype. This slide shows you drug level, what they call log metabolic ratio. So the parent drug over the metabolite expressed as a log ratio. And you can see that this has two groups of people are seen, those with very high log ratio as well as those with lower ratio. And this suggests there are some people who are unable to convert parent drug into the metabolite. And when you have very high levels of parent drug, that would then have very high activity against blood pressure. So no wonder those subjects felt quite faint and their blood pressure plummeted. CYP2D6 has a lot of genetic variations. It's actually quite an interesting gene in that, like I told you, 1 in 11 of you have no CYP2D6 activity, but you're doing just fine. So it is not essential for our day-to-day lives, obviously not essential for exposure to day-to-day environmental chemicals, but it is essential for some of the drugs that we take, because many of you would have severe side effects. And interestingly, what is not shown in this slide is that CYP2D6 also has a gene duplication event. So it is thought that there are people with anywhere up from 3 to 13 extra copies of CYP2D6 enzyme, and in those subjects, you are actually what they call ultra-rapid metabolizers. You're able to really break down drugs very quickly, and in those subjects, you would not gain any benefit from a normal dose of drugs. So you would have to actually give them super high doses to get the same blood level. So this is a little bit of a sort of a list that outlines some of the pros and cons. So when you have a defect in 2D6, you're at risk for adverse effects for some of the drugs that are listed over there. And you're also at a risk for decreased effect for drugs that require bioactivation. So drugs like enconide, as well as codeine, which we talked about before. So I'm going to talk a little bit about some of our group's recent studies in pharmacogenetics to give you a sense as to other things we're interested in. So in addition to metabolizing enzymes, we've been quite interested in what is known as drug transport proteins. So you can see in this picture, there are these proteins that actually help to bring drugs into the cell, shown in blue, as well as out of the body, shown in purple. So this is a picture of a hepatocyte and the blood vessels that feed the hepatocyte in the liver. The liver, as you know, is extremely good at getting molecules into the liver, breaking them down, and getting rid of it in the bile. But a lot of drugs are actually brought into the liver by these drug transporters and then pumped out by these drug transporters. One class of transporter proteins we've been very interested in are these class of organic anion-transporting polypeptides, or OATPs. The table is not essential for you to really memorize, other than to note that some of the important classes of drugs, such as the HMG-CoA reductase inhibitors or statins, are in fact the substrates of these transporters that are often expressed exclusively in liver. What you may or may not know, the statins that we prescribe today for lowering your cholesterol level, its target is actually the liver. The HMG-CoA reductase that lives inside the hepatocyte is the target for some of these statins. So that in fact, what has happened is the statins that are currently marketed and available, the companies have actually, without knowing that they're using the drug transporter, developed the drugs that actually use the transporter to have very high liver to plasma ratio. Because this is a drug you want in the liver, but not in blood as much. Because when your blood level goes up, you have a risk for this very severe and life-threatening side effect called rhabdomyolysis, or muscle injury. Every time you see sort of a TV commercial for some of these statins, they always say, well, if you have muscle aches or pain, please see your doctor or stop taking these statins. That's because when you have high levels of this in the blood, it's bad for you. When you have good levels of these drugs in the liver, it's good for you because then it stops the enzyme that helps to make cholesterol, so your cholesterol level goes down. So let me talk a little bit about SNPs, or single nucleotide polymorphisms, and transporter that we've been studying called OATP1B1. So nearly 7 years ago now, we actually went on a polymorphism discovery mode and looked at populations of people to see if there are genetic variations in this transporter. And there were a number of variations that were identified. Most of them were actually quite rare, but some turned out to be quite common and clinically important. So in fact, many groups have gone on using our information to look at clinical populations, and we now see that one of the polymorphisms that we had reported has turned out to be very important in predicting drug levels. So for example, most of the statins seems to have higher exposure in terms of plasma level if you have this genetic variation in OATP1B1. And also, one of the things that these type of studies are informing us is that, in fact, there may be genetic differences among different ethnicities that may actually also affect the drug therapies. So this is kind of a data that shows you what we think happens in Asians versus Caucasians for a lipid-lowering drug called rosuvastatin, or Crestor. It's a very efficacious medication. In North America, it is approved at the dose of up to 40 milligrams a day, because over 40 milligrams, there's some concern there may be unnecessary toxicity. In Japan, actually, it's only approved for up to 20 milligrams a day. That is because clinical data have shown that in Asians, blood levels achieved is double that of Caucasians. So rightly so, they did not see why you would give 40 milligrams in Asians, because that would be equivalent to giving 80 milligrams in North America, which would not be done in North America. So it does remind us, certainly in countries like Canada and the US, where there are admixtures of many different ethnicities, that in fact, by prescribing the generally recommended medication, not even at an individual, but even as a population or ethnicities, we may still have some concerns or issues, because you can see that, in fact, if you're an Asian, you should get half the dose. We actually carried out some studies ourselves in healthy volunteer subjects. This is nearly 100 subjects, about 70 European Americans versus 40 or so African American subjects, given pravastatin. Pravastatin was chosen because, like rosuvastatin, is highly dependent on drug transporters in the liver. It is not subject to significant metabolism, so it's really a good marker for liver transport activity. Again, it's not essential that you figure out this slide per se, but what we want to impart with you is that a single genetic variation in this OATP transporter alone could be a significant predictor of differences in plasma level for pravastatin. We actually noticed another interesting trend, that because this was, I think, the only or one of the only papers that we actually studied African Americans, we noticed that, in fact, pravastatin level in African Americans is actually lower than European Americans. So it suggests to us that, in fact, the Caucasian population has an intermediate exposure, African Americans have the lowest exposure, and Asian Americans, or Asians in general, have the highest exposure to statin class of drugs. So again, it does remind us perhaps we should think about maybe the African American population need a little higher dose of a statin agent, and Asian population may need a lower dose. I'll just give you a little bit more data onto another transporter, a little bit more difficult to go over fairly quickly, but I'll just teach you a little bit about another protein called BCRP. So this is what we call an efflux transporter. So we know a lot of drugs that we prescribe today aren't fully absorbed. So we think that's because somehow it's just not dissolving well, or it just goes through the body and it's just not going to get absorbed by the intestine. What we may be underestimating is that, in fact, the intestine and various organs have these transporters called efflux transporters that actually prevent drug absorption. So let me go over with you the mechanism action of these efflux transporters, including this one called BCRP. So if you have the drug in the intestinal lumen, this is what the intestinal villus would look like. Here's the drug in the lumen near the enterocyte. If you could focus on one intestinal enterocyte, you'll see the drug will actually get into the enterocyte. But these type of efflux transporters, such as BCRP, are expressed exclusively on the brush border or the apical domain of intestinal enterocytes, and actually actively extrudes drug back into the luminal side, thereby preventing drug absorption. So a lot of drugs that actually don't have what they call complete oral bioavailability, it's not because they're not just dissolvable, it's because they're actually substrates of these efflux transporters that pump the drugs back into the luminal side. And of course, these efflux transporters also exist in organs such as the liver, and actually it's expressed again on the canalicular or the apical side and pumps drugs into bile. So evolutionarily, these type of transport proteins were designed to look for toxins that are in our diet or in the environment to prevent absorption. But if some of it does get through, these transporters in the liver and the kidney would help to excrete them efficiently so that the rest of the body is not exposed to potential toxins. And many drugs that are currently used to the body, it looks like a toxin. So it is treated in a way to more efficiently eliminate from the body. So BCRP has actually turned out to be very important in that many cancer drugs are substrates of BCRP. And we know many cancer drugs actually cannot be given orally, not just because it might cause intestinal injury, but it's just simply not absorbed. And this is in part because these type of transporters are actually very efficient at preventing absorption. So more recently, we actually started assessing for transporter activity in human subjects. So with BCRP, a lot of the drugs that are transported are actually anti-cancer drugs. It's very difficult to study in healthy volunteer subjects, but we became aware of this old drug called sulfasalazine, which is still widely used for inflammatory bowel disease and sometimes in rheumatoid arthritis. Previous work had shown that sulfasalazine, when you give it to BCRP deficient mice, the blood levels are approximately 100 times higher than in wild-type mice, suggesting this transport is profoundly important in absorption of sulfasalazine. So we wanted to use sulfasalazine as a probe drug of BCRP activity, and we actually know there are a couple of commonly occurring genetic variations in BCRP. We genotype these volunteer subjects, and you can see that, in fact, depending on your genotype, your plasma level for sulfasalazine can be markedly higher. So we think that, again, genetic variation in these pathways are very important. And now you can actually start to use sulfasalazine as a marker for BCRP activity and perhaps get a sense as to what BCRP activity is prior to giving chemotherapy to patients of drugs that are substrates to BCRP. So I've given you a little bit of definitions, a little bit of sense as to what we do, but I think one of the goals, or one of the goals for this afternoon is to really ask this question, personalized medicine. Everybody talks about this. I think everybody feels like they have a connection or expertise in this area, as well as everybody wants to make this real. But where is it really going? Personalized medicine has been verbalized in many ways, and a lot of people take credit for this type of a statement. I don't know if any one individual has a patent on this. It's been verbalized as the right dose of the right drug for the right patient at the right time. So personalized medicine is really starting. A number of groups, for example, this is a Mayo Clinic that says they're going to have a database of everybody's genetic makeup as part of their history and the medical records. Countries are jumping on this. So this is saying Japan is getting into personalized medicine, and they're going to focus on some of the drugs that are known to cause toxicity, such as warfarin, which we'll talk about a little bit more, and an anti-cancer drug used to treat colon cancer called irinotecan. So let me go over with you just a brief example of warfarin and blood thinning. Warfarin is a drug that was actually discovered, I think, in the 1930s and 40s. It actually is a serendipitous discovery because according to all the stories I've been told, a farmer in Wisconsin noticed a bunch of his cows had died, and apparently died of what looks like hemorrhage. And I believe he took one of the cows to the University of Wisconsin to some chemist, and they figured the cows ate some sort of clover or some kind of plant in the field that had these coumarols that actually thinned the blood. And then that kind of triggered the chemist thinking, going, oh, we could use this to thin blood. And actually it still is, I believe, widely used as poison for rats, for example. So rat poison has these. But it is still one of the most widely used medication for thinning blood in our patients who require oral blood thinning agent. So warfarin is metabolized by this enzyme called CYP2C9. CYP2C9 has common genetic variations that affect its activity. So this shows you, for example, on the upper panel there, that depending on your genetic makeup, your warfarin dose may differ quite significantly. So why is this important? Because those of you who think about blood thinning or have taken warfarin, nobody tells you about warfarin levels or warfarin dose. When you see your physician, everybody talks about this measure of blood thinning called INR, or International Normalized Ratio, which is a clinical marker of how thin your blood is. So you can imagine, if you had a blood clot in your leg or in your lung, you want your blood clot to dissolve, but you don't want so much anticoagulant activity that you'll start to have bleeding events. So you can see what everybody wants to do is measure this INR and keep people within this ratio of 2 to 3. Why? Because in that range, you have the best anticoagulant effect and the lowest risk for bleeding events. Because you could dissolve your clot in your leg, but if you bleed into your brain, that may not be a good trade-off. So that every hospital, definitely in North America, has this capability of measuring INR because it is that important. And we now know that there are genetic variants in the target for warfarin. So it turns out we knew a lot about how warfarin was metabolized, but we didn't really understand what protein warfarin was acting on. And this is actually a fairly recent paper that was published in Nature by these geneticists that found mutations in this enzyme called VCORC1 that was associated with warfarin resistance. And now other common polymorphisms have been identified that actually shows differences in warfarin effect. So just last fall, I believe, the U.S. Food and Drug Administration approves revised labeling for Coumadin, which is the brand name for warfarin, that genetic tests for 2C9 and VCORC1 is recommended. So is this really feasible? So one of the things we know from published data from various research groups is that, in fact, if you look at drugs that we know are genetically dependent to a degree, you can kind of start to plot this kind of information. So you can see that, in fact, based on your genetic makeup, which are shown in various color codes there, and the dose, which is percent normalized, that, in fact, for many drugs, you should get a very small dose if you are genetically deficient. And in some cases, for example, for CYP2D6, if you have multiple copies of this gene, you should get higher dose than the average recommended dose. And of course, you can see for some drugs such as azathioprine, shown on the right bottom-hand side there, which is related to 6-mercaptopurine, widely used in certain forms of cancers. If you happen to be the unfortunate subject with homozygous for the deficiency state, you should actually get much lower dose than those with wild-type alleles, and this is a huge potential for actually toxicity and death. So let me walk over with you why this kind of an example serves to remind us these type of activities that we want to do in terms of personalized therapies may be worthwhile. So you can see on the top panel, this is looking at a gene called TPMT, or thiopurine methyltransferase, which breaks down 6-mercaptopurine, widely used in cancer chemotherapy. So typically, we would give the same dose of the drug regardless of the genetic makeup, and then when we do this, we'll see profound differences in plasma level. So if you happen to be the unfortunate person who's homozygous for the mutation, you have profoundly higher drug level than those with just a one-copy defect versus subjects that have normal a TPMT gene. And then if you convert that into toxicities, you can see those with the deficiency states would have far greater incidence of toxicity or perhaps even death, because these are highly toxic as well as immunosuppressive agents. You have, of course, high risk of having succumbing to immunosuppression and infection, and maybe even needing things like bone marrow transplantation. So what is, I think, hoped for with personalized medicine is that, in fact, you would be able to do the genetic testing, put you in the right category of the genetic defect. So if you know that you are homozygous for the deficiency, perhaps you should get 1/10 the dose from the very beginning, get half the dose if you're heterozygous or a carrier of one defective allele, and if you're normal, of course, you should get the regular dose. And when you do this, then everyone, regardless of genetic makeup, would have same exposure, because this is what you want. You want to give right amount of drug to the body and to the tumors that would kill the tumor but not the patient. And then if you do this, you would have incidence of toxicity that will be low and equivalent across all groups. So this is kind of the hope in some ways for personalized medicine. So the idea is really to move from what is currently done in terms of prescribing medications with variable efficacy and sometimes toxicity to the future of really kind of a DNA health card in some ways, of identify the right drug, prescribe the right dose, better efficacy, and minimizing adverse effects. So personalized medicine, I think, has challenges as well as opportunities, because not all drugs are as simple or as clear as things like 6-mercaptopurine. So in order to really make research in this area a reality, one of the things I've always kind of laid up front is that, in fact, we need to be able to work together. What do I mean by no more silos? This was an article published in the Globe and Mail in June of last year. That just is an editorial that talks about this problem of insular research or insular medical research. So I guess one of the philanthropists talk about medical researchers operating in very tall vertical silos. So we may become extremely good at what we do, but we're unable to really share our thinking with our neighboring folks. And he goes on to say that there are a lot of discoveries waiting to happen by mixing together of these silos. And in reality, personalized medicine is perhaps such a large and important topic that no single group could completely take ownership and be able to deliver on all of the promises, that it does say to us that we need to work together. In London, University of Western Ontario, London Health Science Centre, we are really focused on our ability to conduct personalized medicine research, and also our ability to study our patients as well as our clinical populations by building this Center for Clinical Investigation and Therapeutics, really to facilitate patient-oriented research and to really support, value, and mentor our clinical investigators, and importantly, to provide core technologies of thinking as well as tools, both in analytical instrumentation as well as genotyping capabilities, as well as things like bioinformatics and statistical support. And importantly, it may or may not be a problem in Waterloo, but in London, we really are trying to foster this notion, while we have many hats that we wear, that we should all think in one unified fashion in terms of the city, and that the resources that we have, whether it's located at one hospital or one medical center, that should be available to all in the city, and that all faculty should benefit. And we will push key technologies, because, of course, just having great thoughts and buildings will not be sufficient when you need certain tool sets. So there are various types of very expensive genotyping platforms that are available. There are some old school genotyping platforms that we still, all of us, I think, still use. So this is a TACMan-based system for genotyping. For sort of low to medium throughput genotyping, this is still sufficient. So you can do 96 genotypes at a time on 96 well plates. So actually, for most academic labs, this may be sufficient. There are now these type of high throughput, large scale genotyping platforms where you can actually create custom SNP chips. You can put hundreds or thousands of SNPs on these chips, and the cost per chip is relatively low, so it may become quite cost efficient to actually genotype for a lot of candidate gene SNPs. And we are pushing hard for the latest generation analytical capabilities. This is, I think, an area that people underestimate in terms of advances in the field. A lot of these machineries are able to measure extremely low levels of drugs and drug metabolites. So within London, we've really made an effort. We are a division within the Department of Medicine, and we've specifically tried to make linkage with our clinical colleagues in various divisions. In addition to that, we've also made specific effort to link up with the various basic science departments as well as other medical disciplines, because I think all of us that treat patients or want to enhance patient care could identify aspects of their research in the area of personalized medicine. And then, of course, one of the reasons for my visit here is really to start to solidify and plan regarding multi-institutional collaborations, and I'm delighted both Dominic and Jake and others here to really start thinking about how we can start forging links into this area that actually expands beyond personalized medicine, what could be viewed as translational research or translational medicine. How can we make our delivery of healthcare to our patients better, and what kind of processes as well as research information can we put to bear that enhances or optimizes the delivery of healthcare. So this is almost the last slide. This is not everyone in our group is in this picture. Some of our folks that work with me in the lab, as well as some of the people that actually do quite a bit of the clinical trials with us. So we are kind of a growing group of people, and I'm extremely fortunate to have great people in our team that actually allows me to sort of go on the road and talk about what we do, take credit for what we do, but really not do much of the work. So if any of you are thinking about kind of a postdoctoral fellowship experience in the lab, I do put this slide in off and on just to kind of reiterate that we are actually a good training ground. So this is one of the postdocs in my lab from about 7, 8 years ago before joining my group, and within about 6 months. So thank you very much.
Well, this is the chance everyone gets to ask questions, make comments, both here and on the web and by video conference. So I welcome your questions. I'm the person that asks the dumb questions. I'm delegated the dumb question answer or asker. If you go back to your beginning slide, way back at the beginning, where you had that one with the caveman, could you go back to that a second?
Sure.
Richard explained something to me today, and he mentioned it again in the talk, but I thought it was so impressive in terms of the thinking behind all of this that I thought it was worthwhile. You managed to put the caveman with the top response to the drug, and the top level actually of drugs, and the others, more recent people, lower. And there's a reason for that. I wonder if you could just talk about that for a minute.
Right. So it wasn't really that I wanted to emphasize our ancestry per se, but to note if we think of really robust, healthy, strong people, I think we have this implicit assumption our external phenotype may be similar to what we do internally. So this would be a wrong assumption. What your liver or your other body parts do in terms of breaking down drugs, their capacity may be completely different than what you might expect just looking at the external phenotype of a person. So this is to iterate that what you think, because that person looks physically strong and would then have the highest ability to handle drugs, in fact, this person would have the greatest potential to actually have a toxic reaction and perhaps faint or even end up in the hospital, compared to what we think are the most susceptible populations, that this child, who you think would be with the most risk for drug toxicity, in fact, may have the most efficient drug metabolizing enzyme in the liver and may be able to get rid of the drug more efficiently than any other group.
The other thing you mentioned at the time was that over time, these we may have had toxic things occur in the body, and the genetic changes that occurred in response to that through evolution may have in fact made us less likely to metabolize that drug now. In other words, it's treated differently due to
right,
evolution.
and that's a good point, because you do start to wonder why there are so much genetic variation and why it's tolerated. So we think some of the genetic variation or differences in frequency of these variations, and possibly in different ethnicities as well as different regions in the world, there may have been evolutionarily pressure to have these genes working efficiently. But as we moved away from that area or started eating different diets, that function was no longer crucial. So, it does kind of go back to some of the discussions we had earlier. Why there are some people with genetic deficiency in 2D6, and there are some people with multiple copies of this gene. And we know actually the multiple copy state is much more common in folks that live in the sub-Saharan Africa. The frequency of gene duplication events are much greater in people who live in that region compared to those who live in the northern climates. So that may have said that perhaps some of our dietary exposures to chemicals that we needed to have in terms of nutrients was associated with potential toxins in the food, and those with ability to detoxify the food but still get the nutrient effect may have had a survival advantage. But then when you moved out of that area that didn't have those kind of toxins, losing this gene made no difference to you in terms of being able to continue, so that then it was permissive in terms of not having these kind of effects. And we see this all the time now with when we do gene knockout mice. So for example, P-glycoprotein, we think it's so important that P-glycoprotein deficient mice have no phenotype until you give them certain drugs. And then they start to have severe toxicity, especially in the brain. And they often seizure and die from normal doses of drugs that you would think nothing would happen.
Interesting. Questions? Do you have some from the web? Maybe we'll take that. Take a second to get a microphone back there.
Question from the web. What do you mean by DNA health card? Does that mean that each patient will receive a DNA analysis that the family physician or expert will be able to access whenever he or she is prescribing medications to that patient? What about environmental factors? Is there any research going on in that category, and if not, why not?
Right, so those are two major questions. I could talk about it for a long time. And within the DNA health card, there's also a lot of issues we need to clarify and understand in terms of confidentiality and access to information and the extent of information you need to make available. So our vision of the DNA health card, we verbalize it as such, but for now it will be focused on drug disposition genes, because I don't know if people really want to know about every genetic variation in their gene that may or may not be associated with disease risk. I don't know if people really want to know you have twofold greater risk for Alzheimer's when you're not 100% sure you're going to get it. Whereas some of those drug disposition gene variations, there's sufficient data to suggest what the impact of it is. And also, it's probably not as disquieting because all of us have these variations, so there's less risk for discrimination or insurance issues or whatever that people worry about. So it would be focused on aspects of certain genes that we would want to study. And then, of course, yes, I think the goal is if we have these type of information, we would like it to be then linked with, obviously, not just the genotyping technologies or information, but also bioinformatics or computer informatics systems that allows us to provide a delivery of information to the physician. So he or she doesn't have to be a super expert in all facets of all the pathway, but to be able to know more than most folks, but still be able to give you expert advice to say, according to the pattern that we see today, it may be best we go with this class of drug for you rather than that class. So we think that may be very useful. And you're right, the environmental differences are a major concern, certainly a focus of our group as well. And this is where, in many cases, we actually may need to measure drug levels. So that's why I told you before, you only have to do genotyping once. So it's actually very cost efficient. But our ability to handle drugs very frequently, so it may be quite useful at the appropriate time to actually be able to measure drug and drug metabolite levels and to see if that, coupled with the genetic information, is more helpful to decision making.
Thank you for your nice talk, and I enjoyed it. And also, I agree with you. A lot of points are quite modern and should be continued. But our feeling was that in the last 5 years, we've tried similar ideas with CHR, but they don't seem to yet see that this is an immediate research need. Do you think the situation has changed in the last few years? And also, second is following question is, because Waterloo is not a medical school, our aim was to collaborate with people outside Waterloo, and we are strong in computational and engineering. And so we try to bring this multitude of information and systems together and come up with solutions or decision-making tools. So we are more from that point of view, we were applying for these grants, but of course, being a non-medical school, I think we were just kind of considered as being relevant.
So that's actually a good point. I think in the past 5 years, there is a shift in paradigm or thinking because partly I think there's a greater now emphasis on what people call translational research or medicine. So I know certainly in the US with our NIH system, they're starting to ask the question, what has medical research done for the patient today? How can we deliver our discoveries into our patients more quickly? And there's been a lot of research dollars being put into these large-scale clinical research enterprises, really, what they refer to as CTSAs, and where they really want to ask everyone to start thinking about applying research that actually can be helpful to our patients. And I think pharmacogenomics and this continuum into personalized medicine is becoming more fundable. Although I think in Canada, it may be that we have not had the critical mass, or perhaps the funding agency have not been as aware of what the possibilities could be. But then the second part is also correct too. It is that not any one of us can completely encompass all the skills necessary to do everything we need, because you're asking for biology, pharmacology, clinical data, as well as output into the patient-physician interaction. So we actually feel that the engineering and informatics component that you guys have is an important asset that should be dovetailed into this type of an enterprise, because otherwise you actually run the risk of having good research packages, perhaps for CIHR type of grants that are focused, but it'll be difficult to deliver on the promise of personalized medicine. So it is a bit of a catch-22 because sometimes when things look like it's really ready for use in patients, CIHR might say, well, where's the hypothesis? It's already working. What we might have to do is really leverage the strength that each group has to say, we have new ways of thinking and mechanistic insight that makes deliver certain care better than what is being delivered today, and perhaps quantify it to say, we save a certain amount of dollars or a number of days in the hospital or deaths or events. And I think applying and merging various technology will be essential for any group to succeed in this area. And we think actually genomic information alone will not do it.
It's a question of my own. And when I'm not helping with webcasts and video and whatnot, I'm involved in a project here with Dr. Chris-Ann DeMarco in the Artificial Intelligence Research Group on highly tailored documentation. And this is kind of a follow-up to that question, in that what we see in the current state of the art of tailored documentation is frankly rather pathetic. Now we're hoping to make some significant contributions and advances and whatnot, and some really interesting things are happening, but I'm wondering about the kind of fragmentation in the sector. And is it a vision, either of yourself or others, that things will somehow gel into a new personalized paradigm and that everything will just all happen? And what are the chances of it happening at once?
Yeah, so I think like most things in the world, it's going to take a bit of an iterative process between various groups. But I do believe the groups that I think have a better sense of the key fundamental things you must have, and then the first steps to make the version 1 successful, will hopefully, should get significant credit. And then if that's linked to deliverables in terms of cost, patient care, hospital stay, lives, whatever it is, then I think it's really a win-win situation. So I think from our point of view, we think deep mechanistic insight, ability to do the clinical studies, and recognizing that additional resources or technologies are required to bring this about is important, because otherwise you'll only do what you do extremely well. And that may, no matter how good your instrumentation is, that may not be sufficient to actually impact the final output, which is better patient care. So we are with you in that we think this informatics, computing, ways of looking at information is important. So we're hoping we'll learn something from you, but we're hoping you'll learn something from us in terms of how we think about drug disposition and response, because I think people underestimate this field in terms of the insight that you need to have. It's not as simple as a single gene causing big effect. We often give examples, and I'm as liable as anyone of showing these big effect problems. But bulk of the drugs that we use today do not use one genetic differences. They use multiple enzymes. And of course, things like drug-drug interactions alone. How do you account for that? So our patients take multiple medicines. You cannot change our people. So we have to adapt our system to make those treatments better, not force them to do what we want because we don't know how to do what they want.
So you could conceivably refer to what you're doing as drug ergonomics, in which a technological product is fit to individual requirements. I guess where I have problems is the technological product, that we know the drug companies are manipulating research results. The journal editors got together a year or so ago and said, we don't want any more of that. So, too often there's a product there that is just too problematic, and the physicians and others don't necessarily know about it. And also, I am concerned, as I think the public is generally, about the side effects generally, going beyond effectiveness. There are just too many side effects. Now, they create downstream opportunities for the drug companies, and that's good for them, but it's not good for the patients. I think maybe, too, we need to follow the lead of nature more. And a number of these drugs are derived from nature, but I think too much of the nature is excluded. If you look at the model of herbal drugs, if you like, that there are factors there that mitigate side effects, it would appear. But those tend to get excluded when a wonderful new patented product is created by the drug companies. And to me, that has to somehow change. Now, maybe it's going to interfere with patenting to some extent if that's done. But I think, where there's a will, there's a way. There's no will to it. I think that the drug companies are taking the easy way, and it leads to technological products that don't sufficiently fit people and cause all sorts of harm, and also increase the demand on the healthcare delivery system, which is potentially bankrupting us, among other things, meaning less money for research at the universities generally.
So I think there's a lot you talked about there, and many important points. It is important, and I think we all recognize this importance of doing research that is really critically peer-reviewed and ability to access data, so forth. You're right. Many clinical journals, including New England, Lancet, many have agreed on data repositories and ability to access clinical trials information. And there is, I think, a degree of rethinking needed, possibly on the part of the pharmaceutical industry about wanting to make one-size-fits-all drug, because companies want to make one drug that can be used to treat everyone, rather than a better understanding the profile within the groups of patients, that some may benefit and some may not, and you may be able to actually take drugs that may have side effects and find out the groups that actually have those side effects and not offer them those drugs, but those that actually benefit and do not have the side effects should get that. So it's actually a different, I think, a business model that may actually fracture their profit a little bit, but in fact, it could also be viewed as an opportunity that you could actually resuscitate a bit of a crappier looking drug on the average, but in some patients, it may be extremely useful. So I think there's a component of that. I think some of the herbal stuff you mentioned there also, with the caveat that also we need a better understanding of how these herbals, preparations, contaminants, all of those things need to be factored in, because I think sure, there is a degree of, I think, perhaps bias about herbal medicines per se, but we also have, I think, a degree of discomfort about not knowing exactly which ingredient in the herbal may be useful and what combinations, if it is combinations, is useful, and then how can we ensure product-to-product safety, and how can we be sure that something grown in this country versus that country. And, we have had a lot of scares about pesticides and adulteration and so forth of herbal medicines because it is not regulated in the way regular medicines are. And perhaps we need to consider that, in fact, it should be regulated and also given kind of a more systematic study. And I think that's all fair game, but this kind of ambiguity that leads to, I think it's actually hurtful to the herbal industry to have the potential for rogue companies that could actually then perhaps make the situation worse for other companies that may actually have standardized product preparation if one company has kind of a poor quality product that is perhaps contaminated and that's doing bad things to people.
I'm actually wondering if you are going to be considering metabolic profiles and possibly even body composition profiles of patients to couple with your work. I'm thinking, I think, more specifically, there's some examples in the cancer literature that have coupled body composition with toxicity of certain drugs. So I'm wondering if you'll be coupling that with your work.
Yeah, so it's, that's actually a little bit of a tougher question. There's a degree of more fundamental knowledge that we need to understand. So there is some thinking to say, and we used to be interested in that area a little bit in terms of BMI and obesity, in terms of differences in capacity for breaking down drugs. There's actually very little done in the field in terms of studying nutritional status, post-surgery, ICU patients, metabolic states, all of those things. And that also, I think, needs to be taken into account. It's a little bit more, it's probably more difficult for CIHR for those kind of grants because it doesn't seem quite relevant. But I think you're right. We know things like diseases and body states affect gene regulation and probably affect our ability to handle drugs, and inflammatory states and other things also affect regulation of gene expression and so forth. So there is kind of an additional set of data that need to be gathered. But at this stage, it's actually quite difficult to kind of come up with a list of metabolic states that you'd like to put on the kind of things you want to do right now without more defined studies.
I actually have two questions that are somewhat unrelated. One is a little bit of an extension of what you were just talking about. Are there studies being done now in terms of the epigenetic sorts of gene regulation issues and identifying markers that may be important there? And the sort of final thing is you alluded to it, and that is privacy issues in terms of a DNA health card. It's interesting. I'm a subscriber to a network that's known as the Rural Med network, which is a conversation between physicians all across Canada. And the last 3 days, somebody brought up the impending new network of lab tests that is going to be available across Ontario. And the issue there is who gets to see the data.
Right. So the first question regarding epigenetics, we agree with you that, in fact, this is also another emerging field, DNA methylation, and also sort of inheritable genetic change, that can be continued on after birth, are actually going to likely to prove to be important. There hasn't been as much work done in this area in terms of drug disposition and response, and I know a lot of our cancer colleagues have focused a lot on epigenetic events and response. So I do think, though, there are aspects of epigenetics that will prove to be very important, and there are technologies that can actually quantify certain aspects of epigenetic changes. And then in terms of, the privacy issue and mechanisms to ensure privacy, it's almost like computers that make sure you log on and you make sure you scramble the website so no one else can hack in. All of those type of issues will become an issue, and we will have to have, I think, very step-by-step discussion. And again, I think Waterloo's perhaps well poised to be able to provide insight into robust systems that can help safeguard information relating to genetic makeup.
Richard, at the risk of going in a completely different direction, one of the factors that you indicated was a factor here was environmental. What are some of the, I'll call it major environmental factors that seem to influence the effectiveness of a drug?
So there are not that many super-duper examples per se, but we do know a lot of processes that regulate gene expression of drug metabolizing enzymes, in fact, not only affect our response to pollutants and cancer risks. So things like dioxin, which has always been studied, actually acts in a very similar way to how I showed you about this gene regulation. It actually activates an AH receptor that turns on some genes. Much of those genes are probably important for endogenous processes, perhaps not so much for drugs, but there are drugs that may be affected by that. But also we know there are a lot of constituents in our diet that may actually also affect our ability to handle drugs. So, I think the environmental things are a little more difficult to quantify for sure because they're often seen at very small levels and also discrete populations where you don't study drug disposition. So it would be difficult to give a yes or no answer, but we know dietary constituents, for example, things like grapefruit juice that's been widely studied, actually can have a profound impact on how we handle drugs. So that is an area that is probably understudied and could potentially give you a significant insight as to why we differ. And actually, if you go deep in this area, there's actually this field called chronopharmacology. In fact, our liver and our body actually may have a sort of a rhythm during the day that our ability to metabolize drugs actually may vary a little bit from when we go to bed versus when we wake up in the morning. So that's because some of the genes that are master regulators of gene expression are also maybe regulated by these clock genes that actually regulate our sleep-awake cycle. So there is multiple layers of complexity. It may be quite difficult to tie it all in at the first goal, but because of complexity, I don't think we should say, we'll just do genotyping and just take the easiest ones. I think we do need to systematically tackle areas that are important and build on component. So really ask the question, what is the contribution of this environmental influence on this set of drugs or this drug, and go through this iterative process for individual drugs as well as classes of drugs, so that we may find for certain class of drugs, certain environmental influences are profoundly important, whereas it may be never important for other drugs.
Now, just a final question is, you're doing incredible work in mass spectroscopy in detecting very, very low levels of metabolites. Can you detect with your methods the presence of, for example, dioxin or other agents, or is that in fact well below?
There's a separate field altogether in terms of toxicology and environmental sciences that people do a lot of these type of work, especially not just in blood, but also where tissues that deposit these things in fats and other organs. So there are a wealth of expertise in some ways, and there are, not that we do them, there are additional technologies in high field nuclear magnetic resonance, or NMR, that actually can detect thousands of chemicals or signatures from biological fluids. So there are tool sets that can get a lot of information, but we have signals for so many chemicals. To know what they mean and when it is important is still a very important problem for us.
But you might know it was present. The reason I ask this is we have a very, very strong collection of people here, a large number of people involved in various aspects of toxicology. Water toxicology.
Right, and there are people also at the University of Western Ontario that are toxicology, biochemistry type of background that also have significant interest, and also concerned about people living in certain areas of presumed environmental pollution or damage.
Excellent. Well, thank you. This has been fascinating as a day and as a talk, and thank you very much for coming here.
Well, thank you.
Well, before you go, I'd just like to give you a small token of our appreciation, and thank you so much for coming, and I hope we can get back together soon. And if you have any further questions, please don't hesitate to send them to Dominic or myself, and we'll try to pass them on to Dr. Kim for his consideration. So thank you for coming, and I hope to see you next month.